EBSCOR, the experimental program to stimulate competitive research, is a National Science Foundation grant program. The goal of EBSCOR is to strengthen science and engineering research and education throughout the United States, and to enable states like Wyoming to be nationally competitive in these areas. EBSCOR aims to improve scientific infrastructure in Wyoming and build science, technology, engineering, and math research capacity across the state. One way Wyoming EBSCOR helps strengthen science research and education is through student programs such as the Summer Research Apprentice Program, also known as SRAP. SRAP is an intensive 6-week paid research experience for high school sophomores, juniors, and seniors aiming to attend college. It offers students a hands-on research opportunity in science labs across the University of Wyoming campus. Students work one-on-one with a faculty mentor throughout their time in the program. SRAP not only provides students with a paid internship and real research experience, but also helps prepare them for college life.
We have Mr. Marten Baur from Lander, Wyoming. He will be presenting water potential measurements for understanding plant stress.
Good afternoon. My name is Marten Baur. I've been working in the Department of Botany under Dr. Brent E. Ewers, and my project is entitled Water Potential for Understanding Plant Stress. So a large part of my project has to do with ecology. And what ecologists do, they study interactions between plants, animals, and the environment. And how ecologists impact the world is that they create a lot of models in order to make predictions. And in order to make models, a lot of excursions into the field are necessary to achieve or receive data. And from this data, such as temperature of an ecosystem, CO2, or incoming precipitation, they can make predictions of inevitable problems or dilemmas that will occur in the future. So, and also a large part of my project has to do with plant drought survivability and drought-induced plant stress. And soil water potential drastically influences this. So in the Wyoming area undergoes a lot of drought. And there's actually two kinds of drought. There's soil and atmospheric drought. This diagram here shows soil, dry soil, and when soil and atmosphere are dry, they actually have a negative pressure. This acts as a suction. So in the atmosphere, the negative pressure sucks the water out from the plant's xylem through the stomata of the plant's canopy. This actually pulls that water. That's how the water gets up to the canopy in the first place, is through transpiration. But the plant wants to retain as much of that water as it can. So in the atmosphere, there's this tension, and the plant's trying to retain it, and the atmosphere is trying to absorb it. And that also occurs in the soil, because the soil is so dry that it tries to suck the water from the roots. So you have this tension from the canopy in the atmosphere, and tension from the soil and the roots. And what this tension does eventually is that an air bubble is sucked into the plant's xylem. And here are two examples of Xylem tubes. Here's an angiosperm xylem. And if you look closely, there's these tiny little circles in here. These are the pit membranes of the xylem. And that is what the air bubble that gets sucked in flows into these pit membranes. And the air bubble actually causes them to cavitate, which this means to rupture the Xylem tubes. And therefore, once the xylem has cavitated, the plant can no longer transport water as well. Going on to Stomata Regulation. There's actually, plants differ by determining whether or not they should open their stomata or leave it closed. They need it open in order to receive necessary materials such as sunlight and CO2 in order to perform photosynthetic operations. But they also want to keep it closed in order to preserve water. And this definitely comes into account during drought. But what also comes into account is soil water retention. Seen here is water, which is completely saturated with water. This is when the water cannot hold on, that the soil cannot hold on to all of the water, and some of it drifts down into the waterways below. Right here is the field capacity of soil. Usually about 30% of the soil consists of water when it's at its field capacity. And we're studying with water potential how well the soil can retain to that water without it evaporating out. So my statement of my topic has to do with water potential and its effects on plant stress and xylem cavitation. My objectives of this project were to create accurate soil water release curves, create accurate xylem vulnerability curves, and to understand soil water potential and plant stress. The field I visited was the glacial till sites, northwest of Centennial. As you can see here, with my arrows here, and here are glacial till sites, where both the we retrieved. We retrieved our soil samples by using a soil core driller, and we've got received depths of 0 to 15, as well as 15 to 30 centimeter samples. And we also retrieved our branch samples from Engelmann spruce trees. This site is called the glacial till site because in the past, a large glacier was actually bulldozing through this valley, and when it hits that soil, it tills it and mixes up the soil, and it actually changes the characteristics. And then we're seeing, by reading water potential readings, we're seeing how this glacial till affects the soil water potential. We gathered samples from PD1, BL1, and PB1, which I'll refer to later. Some of our methods for retrieving vulnerability curves was using a high-resolution flow meter. And a vulnerability curve is a plot depicting negative pressure on the xylem on the x-axis, and on the y-axis it will depict loss of conductivity in the xylem. Once it has 100% loss of conductivity, we can determine that 100% of the Xylem tubes of the plant have cavitated. And this machine tests cavitation because we insert the stem that we retrieve from the glacial sites right here. The root end goes here, and this canopy end goes here to simulate the actual flow of water through the xylem. We use the pressure of just simple gravity flowing through this into the speaker in order to record the gram amount of water passing through the xylem, and by this we're measuring the flow of the xylem. And once we do that, we record those numbers and we place that stem into a centrifuge, which is a machine that spins the sample at rapid rotations per minute. And specific rotations per minute actually simulate drought stress. Because if you're ever driving in a car and you turn very sharply, you'll notice you'll get thrown out the opposite direction. The same thing happens when a plant finds a negative pressure in the atmosphere and negative pressure in the soil. You're getting pulled by both directions, the same as you would get in a centrifuge. It's pulling on both sides of the xylem by spinning around so fast. And therefore, eventually cavitation will occur within that centrifuge. After spinning in the centrifuge, we place it back in here and once again measure the flow. Eventually that flow will render 0, meaning that 100% of the plant's xylem have cavitated, and therefore the plant would inevitably die of hydraulic failure. In order to attain our soil water release curves, we utilize the WP4 dew point potentiometer. It's the machine that uses the method of psychrometry through the clouded mirror, or the chilled mirror dew point approach. And so we insert our soil from the glacial till site within this small chamber, and there's a small space above that sample where water is free to evaporate up, and the WP4 actually chills the mirror with an air conditioning system, and that cold mirror and the water vapor condenses onto that cold mirror, and the WP4 records the temperature of the sample and the temperature at which the water condensed onto that mirror. And it measures the difference between those two temperatures in order to give us a water potential. Okay, so a water release curve depicts soil water potential on the bottom, which the WP4 tells us. And by using equations and the weight of the sample, we can figure out the gravimetric soil water content. Gravimetric soil water content is if you imagine a box like such as here. This is all soil. Then you have like 50% solid soil particles, and then within that solid soil you have air pockets as well as water particles between each soil particle. So at field capacity, soils usually render a percent gravimetric water content of 30% or so. And as you can see here, that's what we have at 30% water content. And this vulnerability curve from Engelmann spruce trees, as you notice, it's from 3 different areas: Bull Lake, Pineville, and Pretty Bull area. PD1 refers to Pineville, so I'll first talk about the red square line here. And these two graphs we made these two graphs because they're comparable. The water potential of the soil, the negative pressure, directly refers how much xylem pressure is on that plant. So we can depict how much tension is going on the Engelmann spruce just by viewing the soil. And we can see at -1, water potential of the gravimetric soil water content is about 8%. When we bring it over here, the negative megapascal of -1, and up to that red square, we notice that when the soil is at the negative pressure of -1, the plant has lost about 70% of its conductivity. 70% of its xylem capacity. And PD1 is a clay soil, and the difference between the soils is that if you have a clay soil and say a rocky soil or sandy soil, the clay soil has a lot smaller particles and a lot smaller pore space between each particle. So when it rains, the clay soil is able to retain that water much better. It doesn't evaporate readily, while as if you have but it also makes the plant exert more energy to pull out that water because it retains that water so well. But if you have a rockier soil or a sandier soil, when it rains, the rain is more readily released through evaporation, and the plant can more easily access it. So during drought, this definitely is a crucial thing to look at, because if there's a 2-week drought period, the clay soil will be able to hold on to that precipitation and therefore have a supply of water for that plant to access. While if you have a grainier, coarser soil, that plant may not have water for the duration of about 2 weeks. So our next area is actually a TB2, which refers to free bowl, like green triangle, and it's a rockier soil. And the percent gravimetric soil water content at -1 megapascal is about 10%. We take the average of those points. And this means that rockier soil, it needs more water in order to give a smaller negative pressure. It's not as water efficient as that clay soil. So when the soil water potential of the rockier soil is a -1, it has a -1 megapascal on the plant's xylem. And we notice because the rocky soil isn't as efficient as that clay soil, there's a greater loss of conductivity in this pre-bowl site, over 80% of the xylem cavitated. Finally, we move on to our EL1 area, our last area, referring to Bull Lake, that blue line over here. It's both a mixture of clay and rocky soil. As you can see, when the soil water potential reads a -1 pressure of megapascals, the gravimetric soil content is about 9%. So, and if we bring that -1 over once again to this vulnerability curve, the -1, it only renders a 50% loss of conductance in that xylem. So by comparing these vulnerability curves to soil release curves, we can determine that the Xylem spruce seem to be doing the best in this type of soil. Our project was limited by time, so we're not able to receive enough soil samples to get a better curves. So therefore, we could have possibly found better correlations between the two graphs. But we did what we could with the time we have. So the applicability of finding vulnerability curves in soil water retention can refer to ecology once again, making predictions about which plants will die during drought and which plants will survive. This also refers to beetle endemics, because pine trees, when undergoing drought stress, they aren't as able to defend themselves against the pine beetles as well. Usually, if a plant is not undergoing a large amount of tension, the plant will release this resin and kick these beetles out. But this picture here depicts these pine trees with the red needles, which occurs a year after a beetle endemic invasion of peat trees, and that means they're dying right now, or they are dead. Forest managements can use soil water release curves and vulnerability curves in order to make models consisting of incoming precipitation and also soil water release curves, seeing how long that soil will retain that water that came through precipitation. And by doing so, they can figure out where the plants are undergoing the most drought stress, and therefore where most plants are dying, and therefore heavier amounts of dry timber. So with that information, they can determine which areas are most prone to fire. And finally, farmers can use this information to reduce water and conserve water, because if they're watering a field and they take a megapascal reading of their water, and if they see it's at field capacity, then there's no point in watering that area more. Rather, if it has a high negative pressure, they realize that their crops are endangered and they need to get water to those areas. Finally, they can know which plants are suited to which soil, and therefore plant the appropriate crops in the location they're living. So by using soil water moisture release curves and vulnerability curves, we can help solve and predict a variety of problems. I'd like to acknowledge the National Science Foundation and EPSCoR for funding the SRAP program. I'd also like to thank my professor, Dr. Brent Ewers, and my mentors, Carmela Guadagno, David Reed, Scott Peckham, Heather, and especially Tim Aston, who has so much patience with us and assisted us more than really anyone else. Finally, I'd like to thank my undergrad students and grad students in the botany and ecology department, my awesome lab partner, Danny Tran, and the SRAP program, staff, students, and for just making this experience unforgettable and making it a really good time for me. These are my references. Are there any questions?